Hello and welcome to the Microscope Lab. This short little video is helpful in two ways. It's going to give you the necessary information about the microscope parts and also show you how to use a microscope in the lab to focus a slide. So what I would like to do is to show you how to get the microscope from the bench and put it on your work area in the proper way. And I'm going to be talking while I'm doing that, so pay close attention. So you're going to stand up, walk over to where we keep the microscopes, and you're going to carry your microscope with both hands. Take it on the bottom of the base and carefully carry it over to your workbench and gently put it down on the table. Now before we switch on the microscope, let's look at the parts of the microscope. When you have the model right beside you like this, it is easy to see the different components of the microscope. Let's start at the top. Up here you see two oculars or eyepieces. This is what we look through when we are studying a specimen on the microscope. Then attached to these eyepieces you see this area. This really just is the extension of the eyepiece. And then up here you'll see that there is a sort of a head area. Towards this direction here going down is an arm. Now this arm can also be used to carry the microscope, but again if you carry it on the arm then one hand has to be under the base. If you look here with me, you'll see that the microscope has some other lenses here and they can rotate if you turn this top knob. This top knob is actually called the nose piece, and it can rotate each of these lenses in place. Each of these lenses are called objective lenses. Here we have the stage of the microscope with a stage clip, and this is how you open the stage clip to put the slide in. Then we have the light source. Look down here at the bottom of your microscope. This is the light source. You will have to switch on the microscope, of course, and there is a power switch here in the back. If you press that switch, you can see the light is now transmitted. On the side here of the microscope is a big black knob. This black knob is used to coarse focus the specimen. And this blue knob inside of it is the fine focus knob. So this is for fine focus. Now, I think the best way to actually show you how to focus a slide is to get a slide from our slide box in the cabinet and focus one together. So go get the box. In your cabinet, you should have a blue box of microscope slides that says Lab 2, the microscope. And there are also little rulers. You can take them out. You will use them during this lab as well. The first slide you're going to be asked to focus in this lab is a slide that has a letter E on it. So if you look here carefully with me, you'll see that there is a tiny letter E printed on a piece of paper and it has been pasted on the slide. You're going to put this E the right way up on your microscope. So I actually flipped it. If I read it now here, I can see the E is the right way up. And I'm going to open this stage clamp and put my slide on the microscope. And then I'm going to use this stage knob. Now I'm going to turn the microscope slightly because there is another lever here, down here, called the rotating knob that you use to move the slide left and right. There you can see it's moving left and right and also back and forth. So you're going to move that until you have the light shine through that little letter E on the slide. All right, so let's look through our microscope. Let's put our eyes close to the oculars, but we're not going to let our eye touch the ocular. Now this is going to take some practice, and what you can also do is you can rotate these oculars. You can move one separately from the other and focus because you know that your eye probably is not the same. The left eye can differ from the right eye. And then use the stage knob again and move that letter E all the way to the center of your field of view. Now the field of view is the circle that you can see when you look through these oculars. And you probably will start off and you won't see just one circle. So this is what you can do as well. You can swivel these oculars up and down. And when you do that, you'll notice that you can see either one or two circles. So you have to swivel them down until you just see one circle of light with the letter E in the middle. And then you follow the instructions in your lab manual. It tells you there what to do with this letter E slide. Now, when you go up in magnification to focus this slide, you always, remember, start at the lowest magnification, low power. Then you go up. You twist the next objective lens in place. Make sure that it snaps in place. You can hear it click when it is snapped in place. And now you only focus with this fine blue knob. You don't touch that coarse knob again. We only use that coarse knob at the beginning. You fine focus for your eyes, and you'll notice that you now have enlarged the letter E. You can see it is bigger, and it probably has shifted a little bit, so you have to go back to your rotating knob and move the E into the middle of the field of view. And again, if you want to go up to high power magnification, 40x, then you twist that one in place. Again, only fine focus, and then you will only see a small little piece of the letter E. You probably will not see the whole letter anymore. The next slide we're going to focus is a slide that has some colored threads on it. Let me show you how to remove this slide so that you can move on to the next slide. We always rotate back to the start. That little tiny lens says 4x on it, the tiny low power one. Then you open up the slide clamp and you remove the slide carefully. Put it back in the box and go ahead and take a colored thread slide and put it on your microscope. Again, move the threads to the middle of your field of view, look through, and now you should see a yellow, blue and red thread right there in the middle of your field of view. Again, if it's not in focus, start up here and focus these oculars. Move, swivel them up and down so you see one circle with those threads in focus. And then the focus knob down here, the big black one, the coarse focus knob. Focus it so that you can see the threads in focus. And you'll probably notice that you can see all threads, all three of them, and they are in focus. Now we move up to higher magnification. Move up and you find focus. So you just look through and you find focus. And your book, your lab manual will ask you to observe which of the colored threads do you see on top. There's one on top, there's one in the middle, and there's one on the bottom. So which one do you see is lying on top? Then it's going to ask you to move to high power magnification, 40x. And there, find focus only again. And you'll notice here you cannot see all the threads in clear focus anymore because you've enlarged them, you've magnified them so many times, right? 400 times. Now, it's important to keep some other things into consideration as well. Whenever you move up to a higher magnification, you'll notice that the darkness is increasing, the brightness is decreasing, so it looks more dark, or sort of faded if you look through the lenses at the image. Now, one way you can compensate for this, of course, is by increasing the light. So look here on the side of your microscope, there's a little wheel, and you can increase the light. You turn it up, so if you look through, you'll see it's brighter, more light will be coming through. Another thing you can do is look down here above the light source. There's a tiny little lever that you can turn left and right. You can move it left and right. And if you do that while looking through, you see that changes the contrast of the image. So that is very helpful as well. When you are all done, ready to put away your microscope, follow the same procedure. Swivel back with your nose piece, take out the slide by opening the clamp, put the slide in the box, turn the light down, switch it off in the back. You can unplug it. Please neatly put the cord together with that little tie. And now you are ready to cover your microscope. And carry it back to its place. Now, another thing that you will also be asked to do during this lab is to measure the field of view, and an easy way to do that is by using a little plastic ruler. Remember, we said you need a translucent one. So follow the instructions given to you by your lab instructor. Put this on your compound light microscope that you just learned how to use. Measure how many, in other words, count how many little lines, little millimeter lines you can see in the field of view, and then you go up in magnification, right? And you'll notice that your field of view will be decreasing. Then, to conclude lab, you will also be looking at a dissecting microscope, which are found against the wall in the cabinets. You can take out 1 or 2 of those per group, and then you can do the same little ruler experiment where you're going to determine the field of view and answer the questions to compare and contrast the compound light microscope with the dissecting microscope. At the end of lab, you'll write a quiz, a 10-point quiz. The quiz will be a picture of this model microscope that you learned to use today in lab, and it is going to be asking you in that quiz to label 10 parts on that microscope. So if you watch this video and you attend lab, then you should have no problem answering those questions. So what I'm going to do is I'm going to move over and focus on this microscope so that you can see the different parts of the